One of the things you're going to notice in cell biology is we talk about some of the techniques that are used to study cells. So there are 4 techniques we're going to go over in this lecture. And I'm not asking you to understand the protocols, so you don't have to know exactly how to do these, like details, but you need to know how they work in general and what they're used for. So the first technique we're going to talk about is immunofluorescence. Anytime you see immuno, I want you to think antibody. And then fluorescence is, think about something that kind of glows in the dark, or in this case, most fluorescents, we shine an ultraviolet light on it and it excites this fluorophore, which is just a molecule that can get activated in UV light and shine. So fluorescence is our detection, or you can think of it as the color that we see. And the use of immunofluorescence is really to detect specific proteins in a cell, in our case. So antibodies, you may have heard of antibodies before. They're what your body makes in response to, say, a pathogen or something foreign. So antibodies are very specific. And they bind to other molecules. So this is just called an antigen here. An antigen is what the antibody recognizes. This could be a type of protein, for example, in the cell. So maybe you want to look at the cytoskeleton, which is made up of proteins, and so you would have an antibody, and the antibody always looks like a little Y. And if you see AB, that means antibody. So in green here we have an antibody and an antibody, and we have some more. So there's a couple different ways to do immunofluorescence. Direct, which is this one here, direct immunofluorescence is when your antibody is tagged with the fluorophore. So your primary antibody, which is the antibody that binds the protein of interest, your primary antibody in direct, how do I want to draw this? Your primary antibody has the fluorescent tag attached. So I call the fluorophore a tag. So it's a way to recognize that antibody. Okay, so direct means you have one antibody that directly binds your antigen, say your protein of interest, and the fluorophore gets excited and you see an image. And I'll show you some of these images in a minute. Indirect, which is the most common, requires 2 antibodies. So you always have a primary that binds the protein of interest. And then in this case you have a secondary antibody that binds the primary antibody, and the secondary antibody has the fluorescent tag. Okay, so in indirect, your signal is coming from a second antibody that is binding to the first antibody that is binding to your antigen or your protein. So that's why it's called indirect. So why would you use one or another? Honestly, indirect is used most commonly because primary antibodies are very specific and very expensive to generate. So if you mark your primary antibody with a fluorophore, you can't use that antibody for anything else except for detection. And there are other uses of antibodies in Western blots, in immunoprecipitations, and other kind of molecular analysis. So a lot of people will have their precious primary antibody just left plain so that they can use it in multiple applications, and then have the secondary antibody marked with the fluorophore that is detected. And you can buy these secondary antibodies much cheaper. This little chart, oh, and I forgot to finish it, shows you that direct has the primary antibody, with the signal, and the indirect has a primary antibody and then a secondary antibody with the signal. So I'll just show you, since I left it here, what this is talking about. So this was saying that you could have a mouse antibody to protein A and a rabbit antibody to protein B. So that means that the antigen was put into mice. The mice made the antibody in this little brown here. And why it's important to know that it's mouse is that here we used a secondary antibody that was goat anti-mouse. That's the brown one here. So that means that we have this brown antibody that was grown up in goats, and it was grown up to recognize mouse antibodies. Okay. So you can see kind of the piling on here. Let's get to the point. Why would you use indirect? Besides cost, your secondary will amplify the signal. So I want you to see here, indirect, when we have A, we have one antibody bound. Now this isn't exactly one, but you can see it's a smaller amount. And in indirect, you can have multiple secondary antibodies binding to your primary. So you get a stronger signal. The signal is amplified. Another reason to use indirect is you can use these same secondary antibodies to detect lots of different primary ones. So in this case, this goat anti-mouse will detect any mouse antibodies you put in there. Okay. And so it's just more universally able to use as a reagent in the lab. What this diagram is also showing is that different antibodies can have different colors. So this is the color of fluorescence. And in this case, they have one that's red and one that's green. Now you have the same here for direct, but the key here is that you're going to get a stronger signal. So what I had said is the primary antibody for molecule B was rabbit anti-B. So they had to use a goat anti-rabbit to recognize it. Right? So just think of this as kind of layers of antibodies. So you can see there's advantages and disadvantages of both. You don't need to understand all of these specifics. I like to give you some context. But the key here is to know the difference between direct and indirect. Direct, the primary antibody is labeled. Indirect, you require two antibodies, and it's the secondary antibody that's labeled. Okay. So we get really beautiful images of cells using immunofluorescence, either direct or indirect. And we usually use a confocal microscope, and this is talked about in the microscopy lecture. versus they call it wide field fluorescence, so it's just not a high resolution. And so I want you to see the difference between wide field here on all the tops and the level of detail you can get with a confocal microscope in conjunction with immunofluorescence. Right? So just amazing detail to look at parts of a cell. And so remember, these different colors, blue, green, red, are binding to different proteins in the cell. So here's just another one of my favorite images. The different types of cytoskeleton are marked in red and green. We will talk in a few chapters about microtubules and microfilaments and intermediate filaments, and those are different types of cytoskeleton that you can detect with immunofluorescence. Right? So you'll have a primary antibody that would detect microtubules and a secondary antibody that would detect microfilaments, and you can get two different colors on there. There's another type of fluorescence that is not immunofluorescence. So see, this is just regular fluorescence. Again, we use a confocal scope, but with immunofluorescence, the cells are dead. Okay? Because you have to be able to stain them. You're basically staining them with antibodies, or you're binding the antibodies to the proteins. And in order to do that, you have to fix the cells, so you have to kill them. Okay. There are some fluorescent proteins like GFP, which stands for green fluorescent protein. And this means you can look at living cells. So we're going to talk about GFP more later this semester, but what's kind of cool about it is you can make your favorite protein and you can genetically at the DNA level fuse it to green fluorescent protein, so it makes what we call a fusion protein. And so that means wherever your favorite protein is expressed, it will glow green. So this doesn't give us the same type of resolution, and we can't do multiple colors with GFP, but we can look at cells, look at some cell structures. You would know where your favorite protein is located in the cell. And I used to use this a lot with living cells, and so we could tag different parts, or we could tag actually viruses and see where the viruses were going in the cell. Continuing on with that, you can use green fluorescent protein in processes called photobleaching and photoactivation. Okay. You'll also see photobleaching called FRAP, so fluorescence recovery after photobleaching. And what's happening here is this cell that's all green would be like one of these cells. Okay. And then you bleach a small area with a laser. So you bleach. Bleach means extinguish or stop the fluorescent molecule. So you kind of damage it so it doesn't fluoresce anymore. And then you watch what happens to this space. So the space is the bleach spot. And you kind of watch it get filled in. Right? So you bleached it out, and you can see it becomes more green and more green over time. This is the measure of the fluorescence. Why would we do this? Well, this is a great way to watch how proteins move. Not how, but I should say the rate of protein movement in a cell. So you can see how fast does it take for these molecules to diffuse. And move around in the cell. You can see how proteins are distributed throughout the cell over time. The flip of photobleaching is photoactivation. So here in this cell we have fluorescent molecules that aren't fluorescing until they are activated. So here they're fluorescing all the time. Here you have to wait until they're activated, and activation is a high intensity laser. It activates those molecules with the fluorescent tag, and then you can again watch the rate of diffusion or movement of those molecules in the cell. And you can see here, if you're measuring just this block, you've lost your fluorescence, and then you've gained a bunch of fluorescence, and then it diffuses until it's kind of homogeneous or evenly spaced throughout the cell. So we'll talk about these techniques as we go along in the different chapters. So I want you to kind of be familiar and understand the difference and similarities between photobleaching and photoactivation. There's a couple cool videos you can watch on photobleaching. I think they're only like 2 minutes long. Let's see if I can pull one up. Okay, so I'm just going to show you a little bit.
FRAP, fluorescence redistribution after photobleaching, is used to gain quantitative information.
We'll start again.
FRAP, fluorescence redistribution after photobleaching, is used to gain quantitative information about the dynamic behavior of molecules in the cell. In a FRAP experiment, a high-intensity laser beam is used to irreversibly bleach fluorescent molecules in one region of the cell. Redistribution of the remaining fluorescent molecules is then monitored in both bleached and unbleached regions.
Okay, so what I want you to see is what they were trying to show you is here they're going to zap that part of the cell.
The remaining fluorescent molecule.
Right, so they're all dark. And then the molecules are moving.
Is then monitored in both bleached and unbleached regions of interest over time.
So you can see this rate of diffusion. How fast does that bleached area get filled in? Or on the flip side, if that area was the activated area for photoactivation, you could see how fast these molecules diffuse away from that area.
By analyzing the time course of the redistribution, we can determine the diffusion coefficient and mobile fraction of the molecules. To simplify the photobleaching experiments and data analysis.
So I'll let you watch the rest of this if you're interested in that. Oops. There's another one. And then this photoactivation is, I couldn't find a good video of it, but this is showing you, okay, here's the cell. You can see it before activation. You don't see anything. And then you zap it with the light, and you activate the fluorescence, and then you can see over time the fluorescence moves, and so now this spot, which is probably the nucleus, now the cytoplasm is moving over the nucleus. So you can see in all of these images how an area that didn't have that molecule that was activated now has it. So, photobleaching, photoactivation, they work opposite, but they really tell you the same types of information. Okay, one last technique is called freeze fracture. And freeze fracture, basically what you're doing is you're splitting the lipid bilayer into two leaflets. So you're splitting a membrane, which you hopefully remember is made of a phospholipid bilayer. And bilayer means there's two layers, and you split the membrane into two what we call leaflets. So this is the extracellular leaflet, this is the cytoplasmic. And what you'll see is kind of smoother areas or really bumpy areas. So the purpose of this technique that we are going to focus on is really looking at the density or distribution of proteins in a membrane. So some membranes are going to have few proteins. You don't see a lot of bumps. Okay? And you can imagine here these proteins come out as bumps, depending on which layer they associate with. Okay? You may have a membrane with lots of proteins. And this tells you a little bit about the physiology of those different membranes. So you're getting a little bit more information about membrane structure. It does not tell you specific proteins or types of proteins. It's just more of a yes or no. How much is there? So, for instance, if you remember, this is in B, this is the mitochondria. The mitochondria has an outer membrane and an inner membrane. And hopefully you remember that the inner membrane, should be IM. The inner membrane of the mitochondria, like this blue, is where the electron transport chain happens for cellular respiration. ETC. The electron transport chain is a series of proteins that moves electrons so that we can eventually make ATP. So I want you to see here the inner membrane has a whole bunch of proteins, bumps. The outer membrane has very few, right? Because the outer membrane, it will transport my mind is going blank, pyruvate in for cellular respiration. But the inner membrane is where the electron transport chain happens. So it has a lot more proteins. Right? So you may see smooth ER and rough ER. Rough ER has a lot of ribosomes on it for protein synthesis. Smooth ER doesn't. So there's lots of really cool images. You can kind of see the structure of the Golgi here that you can get from freeze fracture. But what we're concerned about is just visualizing protein in general, protein levels in a membrane. Are there lots of proteins in that membrane or few proteins, and what can that tell you? Okay, those are the basic techniques for Chapter 1, and you can always come back and review this video as we talk about these techniques again later in the semester.